Hey everyone, welcome back to another episode of Genetics. Today, what we're going to go over is bacterial transcription. So basically, how transcription happens. And then we're also going to include some information on the different types of mRNA. So basically, it's going to be a pretty simple section of the chapter. And then in the next episode, we're going to cover eukaryotic genetics, or eukaryotic transcription. And then we're going to cover mRNA modification. But for now, we're going to talk about the different types of mRNA. I will say that this list is not definite, and it can increase in the future. You will definitely see 5 other different types of mRNA or RNA being discovered, as you move on to your career in genetics. But for now, we're going to talk about the 3 original ones, the OGs as I like to call them, the mRNA, tRNA, and rRNA. So the mRNA is, that's the acronym, but it's basically messenger RNA. And this messenger RNA encodes for a protein So it encodes for a protein that will be translated in a ribosome. So that's basically what mRNA is. It's basically just an RNA that basically has the protein sequence in it, and then that protein sequence will be translated in the ribosome. So pretty simple. Most students, get that pretty easily. You have something called transfer RNA or tRNA. And so basically what this is an RNA molecule that carries amino acids to the ribosome and interprets the mRNA codon sequence. So that's basically what all tRNA does. And you can definitely see some pictures if you're not really familiar with it, especially like what it does. But basically all it has, it's an RNA molecule. You attach an amino acid to it, and then as the polypeptide sequence increases in length, those tRNAs will essentially fuse these different amino acids together to create a ever-growing polypeptide chain. Next, you have something called rRNA or ribosomal RNA. So what this does, it's basically RNA that's used to construct the ribosome, the small and large subunit. So basically that's all rRNA is. It's just used to construct the ribosome. Okay? So here we go with the last 4. The last 4 include something called snRNA or a small nuclear RNA. And these 3 in a row are mostly just used for regulation, and they're always found in last 4. They're always found in eukaryotic organisms. snRNA, what we're going to talk about in section 3 of the chapter, video 3. snRNA is used to construct the spliceosome, is used to construct the spliceosome for intron splicing. That's all snRNA does. Here are the two confusing ones, microRNA and siRNA. So with microRNA, basically what's happening here is that you have a RNA sequence that binds to target mRNA. And what it can do is that this target sequence, basically it alters the efficiency of F of translation. So basically what this is basically an RNA sequence that the cell will produce, and that RNA sequence will go into the cell, into the cytosol, and then it will look for a particular RNA sequence, like an mRNA sequence, and it will bind to it, and then it will prevent it from undergoing translation, or it will alter the stability of that mRNA. And the reason why you want to do this is because, you know, when a virus infects a eukaryotic cell, it produces a bunch of mRNA that is used to make more viral particles. So if you have this mechanism to prevent certain RNAs from being expressed, you can prevent an infection from a viral particle. Okay. Then we have something called a siRNA, or a small interfering RNA. So this is RNA, double-stranded RNA. So double-stranded RNA. This is a mechanism that is used, double-stranded RNA that basically that is used with a complex with another protein, and then this complex is going to work together to find certain RNA sequences, and as soon as it finds the RNA sequence, it will cut that infectious RNA into individual pieces. So basically, double-stranded RNA that is used with a complex to find complementary sequences and breaks the complementary mRNA apart. And then the last one, telomerase RNA. This one is mostly used on telomeres, and this RNA is used to elongate the telomeres. So elongates and maintains the telomeres. Okay, and that's basically all the mRNA molecules. Right now what we're going to go over is the mechanism of transcription. The initial part, or called initiation, it's pretty simple. Every gene has three main components to it. So let's say this is our gene right here. So upstream of every gene, you have something called a promoter. And at the end of every gene, or after the untranslated region, you have something called a termination sequence. So this is basically a typical layout of how you're going to see a gene composed out of. And what's happening here in a bacterial transcription process, you have something called a RNA polymerase. And RNA polymerase basically has the exact same function as DNA polymerase. It works the exact same way. It reads the DNA, the template, 3 to 5, and then it makes a new RNA strand 5 to 3 using the exact same polymerase activity that we've talked about in previous episodes. But basically what this enzyme can do is this enzyme has a sigma subunit in the RNA polymerase, and then this sigma subunit is basically going to wedge into the promoter and is going to separate out the double-stranded DNA into single-stranded DNA. And then because of this, now the RNA polymerase can come in and it will start reading the template sequence like usual, going 3 to 5, and then it'll make double-stranded DNA, or it'll make RNA, 5 to 3. So basically that's all initiation. That's the main gist of how it works. The most important enzyme in this process is the sigma subunit. And remember that the sigma subunit is basically going to wedge itself into the promoter. And then as it does this, this will allow RNA polymerase to begin reading the single-stranded template DNA sequence and synthesize a complementary RNA sequence for the gene. Okay? So that's all it does. That's essentially how initiation occurs. Elongation is essentially you continuing reading that template sequence, adding new single-stranded, adding new RNA nucleotides into your growing RNA chain. And then essentially that is it. So basically what's happening here is you're reading the template sequence, and continuously adding RNA nucleotides to the ever-growing RNA molecule. And that's basically all elongation is. It's basically you're continuously adding new RNA nucleotides into the ever-growing RNA molecule. That's basically all it is. And then as soon as you reach the point where the gene has a signal for, we should stop making RNA, there are two different methods for this to happen. You have something called row-dependent termination and something called row-independent termination. So a row protein, so this is a protein, and this protein is basically a scissor that can just cut the mRNA sequence at the right point, and then that's it. So we can think of this, so row cuts the mRNA sequence, or in general RNA sequence, at a certain termination point, at a termination sequence. So it's basically like scissors. Row independent termination, it's a little bit more complicated, but basically, let's say that this is the newly synthesized mRNA molecule here, and this is the original template. So what's going to happen here during this row independent process is you're going to produce something called a hairpin loop, and you're going to produce this hairpin loop on the mRNA molecule. So this hairpin loop is basically going to have a structure like this. So these structures right here are GC rich regions. This is called a variable region up top, and this is another GC rich region here. And then over here at the end you have a poly U sequence. So basically what happens here, if you remember from the introduction to DNA and RNA, GC complementary base pairs basically have 3 different hydrogen bonds that form in between one another. So they're very, very strong compared to A and U. A and U only have 2 hydrogen bonds, so they're much easier to break, whereas GC rich regions are not easier to break, and they're going to form a loop like how you see it over here. So this loop will form because of these GC rich regions. And then this poly U sequence is basically used to this poly U sequence is basically used to have a way how you can detach the mRNA without using an enzyme for this process, because you need less energy to detach it, right? So how does this work? Basically, this stem and loop structure basically creates a friction, a type of a friction that pulls the mRNA molecule off the RNA polymerase, and then you effectively end transcription from there on. And then that's it. Hopefully this video helped you out. And yeah, see you guys in the next one.